Thanks everyone for joining already now. And I'm wondering if you are wondering what you're seeing in the background. What you're actually looking here at is the underwater work of the Upper Isar River in the Bavarian Alps, where we've been almost exactly 1 year ago to perform an extended field survey collaboration with a team from New Zealand who is also here today. That environment just seems to be very natural and very pristine. And the little fish that you've seen are presumably juvenile European grayling. When they grow up a bit, they're gonna look like that. European grayling has been named the Bavarian fish of the year 2026 because you can be very happy today to see them back in the Upper Isar River after decades of impairment through human actions. Human actions are not just an issue at the Upper Isar River, they are a worldwide problem. And I want to showcase that now by taking a short journey in my postdoc environment to California. What you can see here is Chinook salmon, and I was tasked with their habitat restoration during my 2 years of postdoc over there. One of the key reproduction habitats is the Yuba River. Many Chinook are born here, and when they grow up, they're going to swim down to the San Francisco Bay Area in the Pacific Ocean, where they find rich spawning grounds. But at the end of the life cycle, they're going to return back to the Yuba River and other mountain rivers, where they find cool waters and is presumably use gravity where they can lay their eggs. And also, the Yuba looks very pristine and natural over here, with even the deer crossing there in the background. Now, what you also see in the background are these very high bars there. They consist of course gravel and carbon, which are the remainders of hydraulic gold mining activities that started in the mid-19th century in the Yuba catchment. While it can be very dry and hot in Californian summers, it can rain a lot during the winter. And these winter rains eventually washed away all these mining problems down to the Sacramento Valley, where they caused substantial damages in Yuba City and Marysville. Well, that was one issue that occurred then because of one human action. What do you do if you have an issue with one human action? Well, you implement another one. In that case, was the construction of the point dam to retain that mining debris. That dam was finished in 1906, it's approximately 4.3 meters high and 230 meters wide, and represents a substantial migration variation for the Chinook salmon. Quickly after its construction, it turned out that the dam was by far not sufficient to retain all that mining cost. So they did another dam. This time Engelbreit Dam that was finished in 1941, approximately 80 meters high and 350 meters wide. Also in the US, we can always do a bit bigger. They built a couple of decades later New Orleans Dam, which is approximately 200 meters high and 800 meters wide, and ranks as the 5th tallest dam in the United States. Now, what can you do to restore the habitat connectivity that is lost because of these barriers? Because you cannot just remove these dams anymore because they're part of our critical infrastructure. In Bullets Park, it serves for the generation of low carbon emission hydropower and it serves for water resources management. It helps in the winters to cut off flood peaks, and in the summers, it helps to prevent water into the Sacramento Valley. Now, also, the Upper Isar River is affected by two larger dam systems, and we engineered our landscape in Europe also in similar ways, and also heavily by, for example, straightening them. So these straightening actions serve to gain agricultural land surfaces or to improve ship stability. These improvements, at least from a structural perspective, led then to a fragmented aquatic landscape that is also very monotonous, which is a problem because fish need different habitat patches during their different life stages, and also species prefer differently deep and fast-flowing waters. These different preferences of different fish species are known already for a long time now. And today we also acknowledge this is not just its varying status, it is also their variability, their dynamics, as they change during floods, that reshuffle the riverbed and thus maintain aquatic habitats. We also appreciate that it is also water temperature, water quality in a more general sense, and biologic interactions that in fact where we can actually find fish in natural rivers. In the broad sense, we start to grasp the environmental dynamics that are where we can really find fish in natural aquatic landscapes. And it's not only that, it's also individual differences. So if you want to simulate eco-hydraulic patterns and understand habitat in the river, we're facing two major challenges. One is that we're facing an information gap about what different fish species prefer and actually also what individuals prefer. So where can we really find them in the river? And if we want to rebuild the river accordingly, we also rely on computer models that are inherently uncertain and struggle a little bit with capturing the large complexity of landscape processes in rivers. In this lecture, I want to dive a bit deeper into these challenges, make you understand where the challenges come from, by first defining some terms and definition and how we assess habitat suitability. I will then go deeper into the weaknesses of emerging models and what we can still do to improve habitats. In the last part of this lecture, I will go to some, I find, quite exciting new approaches for simulating ecohydraulics. Let's start with some state-of-the-art terms of definition, starting with the definition of what is ecohydraulics. Ecohydraulics can be defined as the study of interactions between flowing water and ecological systems, focusing on how fluid dynamics influence habitat conditions, including the behavior, distribution, and survival of aquatic organisms. And I would also argue the other way around. Fish can leave their footprint on aquatic landscapes. We assess habitat suitability mostly still with habitat suitability curves as a function of flow velocity, water depth, the grain diameter that is present in a river, and of shelter habitat. You see there that we can evaluate habitat suitability as a function of these different parameters, and you see exemplary curves for Antelope salmon and steelhead for the juvenile fry spawning life stages. These habitat curves get plotted as a function of flow velocity and water depth. Where do we get now that information on water depth and flow velocity from? Well, you can either try to measure them, but that is a bit difficult if you want to forecast them in real world. What can we do so? Well, we can try to model it. The mathematically most simple approach and probably most wrong approach to do so is by 1D cross-section averaging. We would assume uniform and steady flow conditions, meaning that flow wouldn't change in time and not in space, which is mostly not the case in nature. If we do so anyways, we can then iteratively determine flow velocity and water depth as a function of roughness and slope. With that information, we can then go in with habitat suitability curves and derive a habitat suitability index for depth, flow velocity, and the grain size, combine them with a product and get to a cross-section habitat suitability index. Multiplied then with the width, we get to a weighted usable width. And then we can multiply that with the representative length of the ridge that we're looking at to get them to a weighted usable area. Now, today we not only have 1D models at hand, we have also 2D models at hand. And with these 2D models, we're getting water depth maps and flow velocity maps, to which we can still apply these habitat suitability curves then to derive habitat suitability rasters that one can then combine, for example, the geometric mean to get to a combined habitat suitability CHSI raster. Then we can define a critical threshold value, above which we consider a pixel value habitat. We sum out all the pixels that are above that critical threshold value, and we get here we multiply that with a pixel area, and we get to a usable habitat area. There are also other ways to do so, or to deal with 2D model data that use fuzzy logics. Now, from the 2D model, we not only get the flow velocity magnitude, we also get the flow velocity directions. And that is very important information, as fish might be able to drift with the flow, but not always be able to swim against it. In California, I not only had a very empathic PI, I also had very smart colleagues. One of them was Kenny Larieux, who used that information to code up an algorithm to understand fish swimming routes and how well they can escape from hydropeaking conditions. The hydropeaking occurs in a river that is subjected to a dam from which water is turbined during peak hours, meaning when the electricity price is high because demand is high, for example, in the morning. Then when the demand drops, also the discharge turbine drops, and so does the discharge in these downstream river regions here. That leads to a sudden drop in the stage here, which leads then to disconnectivity of habitats laterally when many younger fish in particular are dwelling. In California, the big risks then that in these remaining puddles here, these fish are stranding during the hot and dry summer days. And that is not only a problem here, that is also a problem that occurs in other gravity rivers that dry out during this during downramping the discharges in hydropeaking conditions. Now, with that algorithm, Kenny found out that a ramping rate, down ramping rate of discharges after the peak was of more than 7 cubic meters per hour corresponds to a high stranding probability, while when the ramping rate is limited to 5.7 cubic meters per second per hour, the stranding probability was much smaller. These discharge ranges are very much limited here in the Eulerian, but what could be a much more general criterion is that the stage drop in ramping up and down ramping operations should be limited to just 10 centimeters. Now today we not only have 2D models at hand, we also have 3D simulations at hand, and you can see here a vertical slot fish vein modeled by our PhD student Federica Scolari, who's there, with open foam. Now, if we want to apply this 1D or 2D habitat suitability mapping techniques to a 3D model, we will struggle a bit just in the way in the logic one could apply that. But we gain important information in 3D simulations regarding fish passability. And we just plot one of the cross sections here in these pools here. You can see here the flow velocity magnitude in that pool. You can see that there are low flow velocity patches to the left, to the right, where fish might find resting zones after the exhausting passage to the slot. We can also see that the flow velocity is lower at the bottom and at the top of the water surface here. That is all information that we would not get from 1D or 2D model. The 1D model would just show us in one uniform color, and the 2D model would just provide us with colors that one could correct for some friction. But also the 3D model has some major issues there. So that is a Reynolds average model, and we assume it achieved here some steady state because the water surface elevation was not changing anymore, and the mass balance was stable for a long time. But if you look at consecutive time steps, you see that the flow velocity magnitude pattern slightly changed, and that's a pure numerical artifact that is nothing that is somehow physically reasonable. These change patterns become a bit more important if you look here at the slots in the fish rates in the slot over here. So in that one time it seems like the flow velocity is very high. It exceeds here 1.8 meters per second, which might be the maximum swimming strength against which the target fish speed of juvenile trout swim. In the next time set, it looks like that's not a problem at all. On the central panel here, you can see the flow velocity gradient, meaning the spatial change of discharge. The gradient is particularly high in friction zones, in high friction zones. So where do you find these high friction zones? Well, there where there's a high velocity gap close to boundaries that do not move at all. In that case, you have the high velocity zone here in the slot, right? And then we have the non-moving boundaries here at the bottom and at the vertical slots of the fish ray. So water has a certain viscosity. Because of that viscosity, we have friction and we have high velocity gradients. And don't see a big variation between these flow velocity gradient changes between the two consecutive time steps, but the flow velocity gradient can also be considered as a production zone for turbulence that can be measured as a turbulent kinetic energy, TKE here. And if you look at the different patterns here between the TKE patterns here, between these two time steps, you can see that there are quite significant differences. Again, this is just a numerical artifact, And it is important, but it would be important to determine where the fish is going, because many fish do not want to go in these high TKE zones, but they just do not want to be bounced around with many movements, neither during strong winds. So we're lacking that high detailed information in numerical simulations that fish can perceive through their lateral line systems, and they use that also just to navigate and perceive mean velocity patterns in general. So our numerical models that help us to understand us or are intended to help us to understand that are just very much uncertain. But where does that uncertainty come from? Let's take a look at the underlying equations and how we can still improve having even in that light. So behind these numerical simulations of computational fluid dynamics are two fundamental equations that are the Navier-Stokes and the continuity equations. The Navier-Stokes equations do say nothing else than that the local acceleration of the action term equals the pressure gradient plus with diffusion plus a gravity term. The mass balance is nothing else than that water cannot be magically destroyed or appear in a domain of interest. These two equations now are very precise if we want to look at computational fluid dynamics of steady discharges. But what makes it now that we are struggling so much with simulating? So the big problem with the solution to these equations is that we need to start somewhere and at some point in time. So what we're doing to solve these equations is we're discretizing our domain of interest into a computation mesh, at which we solve the equations iteratively for time steps. So we're not getting smooth solutions, but we're getting sparse and stepwise solution, which makes it very difficult for us to understand and simulate turbulence, which has this high frequency motion. So any flow obstacle will cause the occurrence of turbulent eddies in different motional fluids. As long as these eddies here are first larger eddies, and they will then decay into smaller and smaller eddies until they decay into heat. As long as these eddies are so large that you can directly simulate them on the mesh, there is no problem with understanding the magnitude and direction. But at some point, they will become so small that they are necessarily smaller than our grid. That means the decay is happening at some grid scale, which you can actually not resolve, but you can still model it. And the way how we model it is frequently by using a Reynolds decomposition, where we say that the mean flow field is sum of, the total flow field is the sum of the mean velocity plus a fluctuation term. We can then introduce the Reynolds decomposition into the continuity equations and the momentum equations. But then we're getting here a new second order term, which is just a new unknown. So we're having now more unknowns than the equations with very closer problem. What can we do again with that? Well, we model the closures. One of the most popular, though probably most debated models for that is the K-epsilon model, where K represents kinetic energy and epsilon its dissipation rate. These two equation models bring in some empirical randomness in our models and uncertainty. And they're one of the causes for why we need to adjust every numerical model to every specific case studies where we want to use that. This adjustment process is what we refer to as model calibration. A model calibration, what we're doing is we're having some first idea about an uncertain parameter, being a calibration parameter, with which we start our numerical model. Then you run the numerical model and you compare it with measurement data. So we need this measurement data, which represents ground truth, to calibrate our numerical model. Then we adjust these calibration parameters in a way that we think makes the model better emulate the model, the measurements. And we continue that process until you get somewhere where we think at least that the model is now somewhat reliable. Now, what are calibration parameters that we would consider typically? Well, a shallow flows, of course, it would be water roughness. But as we get into the understanding of deeper waters, which is particularly important also to understand habitats of larger fish, there is much more. There's much more to consider, and to understand that, let's take a closer look at the Reynolds average Navier-Stokes equations. That is basically the same equation that we see the two slides ago, just now these orange terms will fall apart under the Gaussian approximation, which means that we can neglect density fluctuations except for buoyancy. That means all these rho prime terms will have fallen apart. And now I want to draw your attention to that term number 7, which is viscous diffusion. There is that mu in here. That mu is a dynamic viscosity. The way how we bring dynamic viscosity into the numerical model is by summing the share of molecular viscosity plus the amount of resolved turbulent viscosity plus an rather trivial amount of subgrid scale viscosity. Now, if we want to understand habitat dynamics, we are also interested into the transfer of scalar water temperature, which we can follow up by solving the advection diffusion equations that contain a similar term, which is diffusivity. The diffusivity is similarly implemented in numerical models as the sum of molecular diffusivity plus the amount of resolved diffusivity plus the subgrid scale mixing diffusivity. So we get in with that here is two calibration parameters in terms of the background eddy viscosity and diffusivity that we may want to calibrate. And to showcase that now by means of the Schwarzenberg reservoir in Black Forest, that is part of a storage hydropower scheme where water is turbined from the reservoir through that intake tower that you can see in the center and pumped and the received water through pumping observations and operations through that same tower. The reservoir also receives water through minor tributaries and a conveyance tunnel. A long time before I got here to the University of Stuttgart, my former colleagues collected data from that reservoir in terms of water temperature profiles and horizontal flow velocity. That data served then for the calibration of a 3D flow model. So the measurement data were here water temperature profiles plus horizontal velocity. And the calibration parameters of that model were the background horizontal eddy viscosity and diffusivity, in addition to the water temperature at the inlet tower. Now we directly get here to the results of that numerical model. You can see here at the left-hand side the water temperature profiles in the red dashed line. That's in comparison here to the measurements here in the black circles. Now here to the right, you can see here the modeled and measured horizontal flow velocity over the simulation time of 6 days. The blue line here represents the model output, and the black line here represents the measurements. So that is a perfect fit, but you can see that trend-wise water temperature profiles and horizontal flow velocity trends are pretty much well reproduced. But I want to draw your attention to these many gray lines in the background and other results of thousands of calibration runs that we used to calibrate a numerical model. For anyone who's a bit familiar with numerical simulations, like Bob and me, did surprise them with 4,000 of calibration runs, and that was possible through a collaboration with Samuel Lagrangian, who was also here, one of the statisticians at our institute. Within the Bayesian calibration framework, we are using surrogate models to bypass the long computing time. In that calibration framework, we have a private belief about our three calibration parameters here that has a uniform probability in between the minimum and the maximum. Then, through that calibration, we're moving to posterior distributions that have a possibly narrow width to highlight us where we can find possibly clear optima. So in that case here, you can see that for the initial temperature at the inlet tower, we had a very clear peak. So very low uncertainty regarding that calibration parameter. But the peaks for horizontal background eddy viscosity and diffusivity are a little bit less clear. Although for the horizontal background eddy viscosity, the value of 0.3 is still rather low and means that we had a pretty good horizontal momentum exchange model. Now for the background horizontal eddy diffusivity, the maximum was at 1.8, which is quite high and points already to that we have an issue here with this scalar diffusivity. And that becomes even clearer if you keep in mind that we are accounting with both parameters of the same conceptual model weakness, which is the processes that are happening in subgrid scaling. So the physically reasonable ratio of these two parameters would be approximately 1. But if you take the ratio of these two parameters, approximately 6. So why is that? Well, first of all, it's important to know that Bayesian calibration is a pure statistically driven method and nothing that follows physics. So if we would have this in human calibration, we would probably not do that because we want a ratio of 1. Now let's try to understand what was happening here. But the Schwarzschild reservoir is subjected to stratification. Stratification in a lake occurs because water has different densities depending on its temperature, with its highest density at 4 degrees Celsius, meaning that in winter we will have the lowest and the highest density values at approximately 4 degrees Celsius at the lake bottom, while we have cooler water layers at the lake top. Now in the summer, the whole thing swaps over and the warmer water resides at the top, where the cooler water resides at the lake bottom. That certification process takes in the Schwarzschild reservoir several weeks to set up. Now, the numerical modeling calibration, we only gave it 6 days to adjust the calibration parameters. And the statistical process found that this was only possible by artificially ramping up scalar diffusivity, which makes sense, and I might still sound a bit enthusiastic about the model results here, although the model itself was not very meaningful. And with that Bayesian calibration technique, we have a tool at hand to troubleshoot issues that are in our numerical model, and we applied that now to different model types that have hydrodynamics around fish, not only, but also sediment and morphodynamics. Before I get now to how we bring sediment morphodynamics into the numerical model, I want to take a short break and ask if there's any questions before I continue. Yeah, thanks for the question. That probably had some to clarify some issues in my explanations. So we started off with a horizontal and vertically constant water temperature profile, which was definitely not present at the model start, but we assumed it was present. And then we gave the model a runtime of 6 days and wanted that after 6 days it shows the water temperature profile of a stratified layer. But that can only happen if you ramp up the diffusivity in a way that's not meaningful. So I will continue with how we bring morphodynamics into numerical models, which is basically mostly done by solving the external equation that says that topographic change or maybe bed elevation change over time is a function of the porosity term plus times sediment transport. The most relevant part of sediment transport here is the moisture that is moving directly on the riverbed by rolling, sliding, and bouncing. However, the rivers, one of the most popular approaches is still the Meyer-Wiener-Müller equations from 1948. That equation has the different terms here that are just here to bring in some dimensions. And then there is that difference between these tau x and that tau x CR, which is dimensional spatial space minus its critical value. Dimensional spatial stress is also referred to as the Schmidt's parameter as a function of water depth, slope, sediment rain water ratio, and inherent certain characteristic rain source. Now, its critical value can be understood if you look at that plot here, what's known as the Schmidt's diagram. That plots here the dimensional spatial stress over the particle Reynolds number. Now, the dashed line here is the critical value of the dimensional spatial stress above which you would expect Bedload transfer to occur. So if you're having a situation in a river where we measure a certain energy slope, a certain water depth, we're having a certain sediment water drain, sediment drain to water ratio, and the characteristic ratio that might fall here or here or here or here. If you fall below that graph, then we would not expect Bedload to occur. If you fall above that graph, we would expect Bedload to occur. So just from the theory here, what we expect, so moving here from the bottom left to the top right in the diagram, Bedload transfer becomes more. Now you can see here some Bedload transport measurements where the marker size scales with the measured intensity. So the larger a marker is, the higher was the measured Bedload transport intensity. And the theory that the Bedload transport intensity increase on both left and right is very well captured in these blue circles. Become bigger as you move from the bottom left to the top right. But it is not very well captured here in the background by these gray rectangles that are more than 12,000 measurements from real rivers. So we're wondering, why is it that in nature we do not see increasing Bedload transport rates with increasing dimensional spatial stress? For that purpose, we ran an extensive data survey with master students to understand local factors that feed into the dimensional spatial theory that you can see here in the top gray box, and in the bottom box you can see the catchment and climate controls. Important to understand here is that correlation matrix that blue corresponds to hyper in blue set is the higher the correlation, and red means anti-correlation with a minimum value of -1. Important for us here is that the first column that is dimension is Bedload compared to all the other factors. What you can see here that there are some parameters from that shear stress theory that explain part of the variability in the Bedload transport measurements, but by far not all. And then even as high, almost as high as share in the variability in the Bedload transport measurement can be found just by catchment and climate controls, like when, in what season was the Bedload transport measurement made. No reaction about the season parameter? Well, that sounds a bit bizarre. It's not a physical process when you do the measurement, but that was related to snowmelt. Whether the snowmelt was active during the Bedload transport measuring or not. So the consequence for us, we want to measure Bedload transport or model it in the vertical model, is that we need to account for catchment or climate controls, at least at the model boundaries, for example, by coupling them with hydrological processes. Now, Bedload is by far not the only contributor to the total load that you can see here plotted over the water color in the center here. There's also another type of sediment transport, which is suspended load of finer particles that follows that belly shape here over the water line, but still larger, smaller particles are moving closer to the river bottom. Compared to suspended load, Bedload makes only 10 to 20% of the total load. In mountain rivers, up to 40%. So that is a minor share, but it is very important for ecosystem maintenance. And for some human actions, we cut off the cause Bedload supply and reduce it to almost zero. What could happen here? If we build a dam, for example, we're going to generate backwater. At the head of the reservoir here, very quickly, the coarse share of sediment will almost immediately deposit, quickly followed by sandier fractions, and only very fine fractions are transported across the reservoir completely. Now, the consequence for downstream reaches is that so-called riverbed clogging can significantly intensify. You can see here the almost completely clogged bed of the Inn River in Germany. So what happened here was that the Inn River has naturally rather coarse sediment matrix. With no coarse sediment coming anymore from the downstream sections, the existing riverbed suddenly became somewhat stiffer, organized itself. So it couldn't be mobilized that quickly anymore. And fine sediment particles started to infiltrate the porous space of that coarse sediment matrix. So the fine sediment would start to deposit here until it was completely filled up, as there was no motion here, no renewal anymore here of these coarse sediment fractions. The consequence of that is that rather oxygen-rich surface water, because of photosynthesis, cannot penetrate anymore into the deeper substrate. And the living space, that niche here in that hyporheic interstitial, almost completely disappeared, which is a problem for macroinvertebrates that are living here. But fish like to forage on macroinvertebrates, so they won't find a lot of food anymore. And the missing oxygen penetration also is a problem. You can see here that illustration here of a preschool that was still pulled out in the river under the supervision of Stefan Horn, who's also sitting here today. We can see that there's almost no oxygen in the deeper layers. Now, that clogging process also led to a condition here that increases here the resistance of the riverbed to any acting forces. So if a gravel-spawning fish tries to dig its spawning raft into a gravel bed, it will struggle to do so. And if it still manages to dig its spawning raft here, it will lay its eggs into that spawning raft, and when these eggs become larvae, the eggs and larvae will struggle to find sufficient oxygen to survive. So clogging is a major issue for ecohydrologically well conditions in a river. We want to possibly restore clogged rivers. Now, what can you do? Well, probably in particular in Europe, you can often not just remove the dam. That would probably also not just solve the important clogging problem as it is already present. And that is still part of our critical infrastructure. But what we can do is to act locally. You can see some examples of local installations. That here is so-called half-moon structure installed at the Inn River, which consists of a rather large cobble and boulder, which is not so natural here for the Inn River. One can also go, as you can see here in the right circle, into the river with an excavator and wash out the fine sediment here. That works for a couple of weeks, maybe a couple of months, until it is clogged again, and we go with an excavator in a water-sensitive ecosystem. Another option here to do is to implant large wood pieces. We know already that large wood is very beneficial for rivers because it modulates hydraulics and morphological patterns. So that helps us to better accommodate the different needs regarding differently fast and deep from the waters of different species at the different ages. Now, we were wondering if one can also use large wood to declog a riverbed. We addressed that question in a seed-funded project in collaboration with Riverbed at MIT and Isabella Schalko at WSL in Switzerland. In that seed-funded project, we ran food surveys in the Inn River and flume experiments in Boston. We installed two large wood pieces in a morphodynamic fishway that bypasses the dam and the inn with the help of Verbund. Thanks for the help here and the permissions here. And then after the installation of the large wood pieces, we measure the interstitial dissolved oxygen supply here upstream and downstream of these large wood pieces in the riverbed. Then Verbund let run a morphologically effective discharge over these large wood pieces. After that flood, we went back into that morphodynamic fishway and we remeasured interstitial dissolved oxygen concentrations. You can see here the result measurements of this interstitial dissolved oxygen concentration from before and after that morphologically effective floods. The profiles with the circles here, left in that case, are measurements from before the morphologically effective floods, and the X profiles are from after the morphologically effective flood. So if such would trigger declogging, we would expect that the circle graphs move to the right to the X graph, which was the case here for the rather gray scale here, where a significant improvement could be observed regarding the interstitial dissolved oxygen concentration, and we even got out here of that critical range that is critical for many organisms to survive. Now we could observe small declogging trends also in other profiles, but they were not so clear, and also from the other large workpiece, the profiles were slightly promising, but also not that clear, although we could see that larger pieces led to a stronger declogging footprint. That's something that we could also confirm in the lab. We basically saw particularly that emergent rocks have higher declogging footprint than small rocks. Now in the lab, we saw that the declogging efficiency was highest at the tip of the log and in the wake of the tip of the log. But in the field, we couldn't get here because our survey equipment is just not able to get here to water depth of more than 0.5 meters per second. So in that seed-funded project, we got a basic proof of concept that large wood can help to trigger declogging. But to really understand how that works, we just submitted a new research proposal together with Ingo to better understand and systematically analyze declogging effects of large wood with the help of field surveys and flume experience. And we also want to get to something that has some predictive capacity. We want to get to some computer modeling framework, numerical simulations, to model potential declogging effects. But that is still a challenge because, remember, we will need a very high resolution to capture all the details. We need to understand eco-hydraulic patterns. That is to a very long computing time. You also remember that if you're simulating sediment transport, we're already having a very high uncertainty in the ground truth. Now, if we want to simulate declogging efficiency, we also need to simulate the exchange of oxygen-rich surface water with the subsurface, which adds then another computational domain, so more computational complexity. Because we also want to observe morphologically effective floods, and we also need to simulate topographic change. Remember the Exner equation. More computational complexity. I'm still quite confident that we can get there, and we're going to see later why. But if we get there, we will probably have a good picture of hydrodynamics, morphodynamics, and potentially other environmental dynamics too. That could be water temperature, turbidity, or just the smell of water. But what we're still missing here is the link between fish and how they interact with the environment, and that brings me back to these two initial challenges of the information gap that we need to understand better where we can really find fish nature, and if we then want to improve habitat conditions, we need to improve numerical models and extend their capacities to be able to capture the whole complexity of that environment. So I hope you're now with me with understanding these two challenges. We can now get to new approaches here. I think finding solutions always starts with acknowledging what you already know. Know that in an aquatic landscape, we're having elements that interact with each other. We're having sediment that is moving through our region of interest and will affect how water can move through it. If we're having large wood pieces that are present in our region of interest, they will affect how sediment moves through the domain and also how water moves through the domain. Now, we're having fish individuals present in our domain of interest, they will also leave their footprint in the aquatic landscape. There was a very interesting study from Marwan Hassan and his colleagues who found signatures in Bedload transport measurements from salmon that were digging their spawning nets in the Riverbed River. So what that means is you rather want to look at the aquatic landscape from an individual perspective rather than just from top down. Just imagine you're planning a hike, I know that makes sense. But you're looking at the map, and then if you're on the site and you're trying to follow up the hiking path, it looks completely different from what I've seen or imagined on the map. Technically, that corresponds to switching from the Eulerian to a Lagrangian perspective. So in the Eulerian perspective, you're standing at a fixed observer position and you're looking down somewhat at our trivial landscape. And that is what produces us from numerical measures, qualitative maps, velocity maps, and suitability maps. But from a Lagrangian point of view, we are diving into that domain, we're following flow paths. One of the first approaches in ecohydraulics that was accounting for that theory was the Eulerian Lagrangian agent method that was first published 20 years ago, where the hydraulics are still solved in Eulerian mesh. And then we have in Lagrangian fish individuals with tracking of sensory query distances that makes that we have found now for fish as agents that have behavioral rules that can be triggered by hydraulic stimuli. Now, the question is, where do we get these behavioral rules from? That brings me to that first question, or that first challenge, what can we do to get that data? And there are two fundamental approaches. One is data assimilation, and the other is knowledge production. So in knowledge assimilation, what we do is we have its observation, and with that observation, we're moving that into some kind of scientific processing. That occurs, for example, if we are observing how well a fish can find the entrance of a fish bypass that is installed somewhere on a river to bypass a river and with that. And if we want to observe also how well fish is able to swim through that fish way, that is also part of this kind of observation to track just that. Another option is to use eco-hydraulics fuels. There's a very prominent example installed in the Bundesanstalt für Wasserverkehr and Environment in Karlsruhe, where they use that fish tracking software to get to something that are individual-based models to better understand where fish really want to go. That is a great approach and these are great results. Now, knowledge for production is a bit more complex. You have not just a one-way direction from observations to scientific processing. It keeps its eyes always on all elements here. A very good example for that was the two-eyed ceiling framework that keeps always one eye on indigenous knowledge production and one eye on scientific knowledge processing. That approach helps them to bridge knowledge action gaps that might occur if you just have a scientific outcome and just blow it out into the world, apply it here, and do something. But we want to get feedback from how well that really performs if something is restored according to that, for example. Now, this bridging of knowledge action gaps not only requires a collaboration between scientists and indigenous communities, also can involve fisheries or any other stakeholder that can provide us with information how good habitat looks like. And with these approaches, we're getting to something where we can better understand environmental dynamics that really determine if fish want to go in or the river. We're still facing one fundamental problem: how to pack all that complexity in a numerical model. Well, there are two very exciting figures of light here, I would say, and they lie in the particle itself, PIC and fluid implicit particle methods. In those methods, we are still discretizing our computational domain into a numerical mesh, but then we're placing fluid parcels into the computational domain. These fluid parcels have their own parcel velocity. We then project the parcel velocity on the numerical mesh where we solve the governing equations. Why do we do that step? Well, because solving equations on the mesh is just very stable and very robust and computationally efficient. Then the PIC update, you reproject the flow velocity on the parcel, so completely replace the parcel velocity, and then we move the parcel with its flow velocity magnitude and its direction through the domain. In the FLIP update, we only replace the velocity of the next time step by the difference of the partial velocity from the last time step and what has been calculated in the mesh. Well, why am I bothering now towards the end with these tiny little details? Well, the result in what you observe in a numerical simulation is very different when you're using PIC and FLIP. PIC is very dissipative, meaning it leads to very diffusive fluid behavior when you observe it. And the FLIP method is very conservative, and it leads to a very noisy and jittering fluid. So if you want to account for diffusive effects in our numerical model and get that possibly right, according to measurement data, we're getting here a calibration law if we're having a PIC-FLIP ratio to adjust how much PIC and FLIP is in a numerical simulation of that type. So we tried that and benchmarked a PIC-FLIP simulation against open form simulations from Fidelity and Fishware. At the bottom, you can see here the PIC-FLIP installation that our master team Barry Kenninger set up. And they both simulated the 100 years flood scenario in that vertical stockfish way. You can see here very high flow velocity patterns that resemble in both simulations. Please keep that the flow velocity is quite high. Then the velocity in the FLIP for the upstream end of the stockfish way are also a little bit higher compared to the lower velocity here at the downstream end in the structure. Now, in the PIC-FLIP simulation, you can put in place a fish avatar, and then we design here that nice little trout here, around which we can then define an interaction radius that enables the fish avatar to interact with the fluid parcels. Let's take a look at how that PIC-FLIP simulation looks like when you put it into action. We can see here the simulation starting with a smaller discharge, zooming here first at the upstream inlet back, and that's where the upstream condition here corresponds to a spawn sphere that is residing in an upstream tank, like you might know it from physical flume experimentation. Then we're going here into an upstream stretch where the parcels are accelerating toward the vertical stockfish, where you're going to see that high flow velocity jet that you could also observe in Fidelity's output from the open foam simulations. Then we go to the downstream end of the vertical stockfish, where you render the parcels into water. You're going to place the fish avatar now in that simulation. We raise the discharge to 100 years, but now swim with the fish against the current, because that is where we want to, why we are simulating that fish wave. Oh, we want to understand if the fish is able to pass through that fish wave. And you can see here the fish is very well able to do so in the downstream stock, where the flow velocity was lower. A 1D or 2D one, the told us that is impossible. Now, if the fish is approaching here the upstream spots, you remember the velocity is getting faster, a fish is repelled by the stronger currents. Now, finally, the fish will make it through the fish way and arrive at the upstream end of the vertical stockfish way. And there it is now caught by these very high velocity that you can hopefully remember. That is a poor perspective here in that case for the fish in the end, but I think this is a very great perspective for ecohydraulic modeling, with which I want to get a conclusion of this lecture that I started with showing you how human actions left legacies in fluvial landscapes. You've seen how the construction of dams can amplify riverbed clogging, and that with large wood we might have a tool at hand to alleviate clogging, and I hope that our project is funded to better understand that. You've also heard that large wood modulates hydraulic and morphological pattern, which is advantageous to both the different needs that different fish species have during their different life stages. What we typically do to assess these different needs and habitat suitability accordingly is to use habitat suitability maps based on 1D or 2D models, although we already have 3D models at hand, to which we can partly apply this habitat suitability mapping approaches. Still, we have seen that numerical simulations themselves have some weaknesses regarding the uncertainty. If we only consider physical quantities, we will still probably have a rather poor understanding of where we can really find fish in nature. To better understand that, the Eulerian Lagrangian agent method was a very important step where fish become Lagrangian agents that are moving through our computational domain. At the end, you've seen here these FLIPs assimilation that basically treat any element in our region of interest as Lagrangian agents. Now, before I close the lecture, I want to leave a big thank you to anyone with whom I had the opportunity to collaborate in the last 10 to 15 years. If that was for discussions, if that was for writing papers, if that was for writing projects, if that was for doing free work together. And I just want to express today, thank you also to all students who were supporting us in these processes, and some of them over here today. And at the bottom left, you see the emblems of funding agencies, and without their financial firepower, it would have been impossible to get to these scientific results. Finally, I also want to leave a thank you to Riverbed to guiding me through that and the implementation process, and to anyone here in the audience for being present. And if you didn't see your picture before, it is just because I couldn't scrape it from the internet. So you're very warmly to send a picture that I won't be able to do that invitation again.